Welcome everyone to our webinar today. Mark Buemiller is going to be talking to us about nanoparticle applications for drug delivery. My name is Ian Treviranus. I'm the product line manager for HORIBA's particle products in the US. Mark is a vice president currently stationed in Europe, and I think today's presentation will probably last, I don't know, what would you say, Mark, 40, 45 minutes?
Oh, I'd say 30, 35. I'm hungry. It's late at night.
You're hungry and hungry. And
it's already 7:37.
We'll have a Q&A session immediately following. So without any further ado, please begin, Mark.
Sure, you forgot one other thing we usually do, Ian.
That's right. Mark, you have a joke for us, I think.
The physics joke. Well, you will determine if it is a joke. These are way too serious. All right, so two atoms are walking by and meet each other on the street. And atom number one says, I left the house today and I think I lost an electron. Atom two says, are you sure? Atom one says, I'm positive.
Ba-dum-bump.
On that note, we will switch to nanoparticles for drug delivery, which I see as a very interesting topic. I actually really enjoy it when I get a chance to go visit customers, and when I see that we sell instruments into this particular application, when I get a chance I like to go visit, just because I think it's one of those areas where there's a lot of research, a lot of good particle technology being applied, and I just find it interesting, so I also like to talk about it. So we're going to talk about nanoparticles. This implies used in the pharmaceutical industry. Why don't we start by saying just why is particle size important in general in the pharmaceutical industry? And I'll start by breaking it into, it's a question of the drug delivery method. If we're talking about tablets, then usually this is a powder, and here the active ingredient, the size of the active ingredient, has an influence on the dissolution rate, also on content uniformity. And if you're taking these powders and pressing them into tablets, what you do with particle size will affect the quality and the hardness of those tablets, partly because both the size and the shape affect the packing. So you can imagine if you take a bunch of tennis balls and try to jam them together, it's very different than taking a lot of hockey pucks. And also, a pharmaceutical plant is just another chemical plant in many ways, and the size and shape of the powders will affect the powder flow, just in general in moving the powder, but also in the unit operations like mixing, pressing, things like that. Now if we're talking about suspensions, then here we have the same concerns of dissolution and content uniformity. But also if those particles are too large, then we could also run into trouble with, I have to escape. So sometimes I get this drawing tool. And I have to go here, and I apologize for the delay here, and then I have to go back to where I can actually show my screen. So what happens is I lose my ability to point on the tool to say show my whole screen.
So click the escape button.
Yeah, I did that.
There, now you've got your pointer back.
And then I get my pointer, then I go to here.
There you go.
And then I get my pointer back here. Sorry for the delay. You know what it is since I'm traveling, I don't have my mouse with me. And I'm just using my keypad, so sorry about that. So did we go back to where this is showing correctly, Ian? Escape again. Hide that. All right, but I lost my pointer. I grabbed my pointer. So note to self, when you're traveling, always bring your mouse with you. So when we're dealing with suspensions, if those particles are very large, then what you do is you just shake these bottles. Usually you're giving a suspension to your child maybe. And if you shake the sample, the active pharmaceutical ingredients, if they're large, heavy particles, they might not suspend quickly, and then you might have trouble with content uniformity again. And also just the feel in the mouth. Very large particles feel kind of strange in your mouth. When I mentioned that particle size affects dissolution, there's a very direct relationship here. What we see in this equation is this is one of the forms of the Noyes-Whitney equation. And what we're saying is dx dt. This is the dissolution rate as it goes from a solid into a solution. As a function of time, various factors influence this, including the drug diffusivity, the initial drug mass, the density, the diffusion layer thickness, and down here R0 is the initial particle radius. So you can imagine large particles take longer to dissolve than small particles. So this is just showing how there's a very direct relationship between the size of the particles and dissolution rate. And what I'm showing on the right is data from the publication I note down here, where they're showing dissolution. This is percent of drug dissolved, how it changes as a function of time. Actual data versus the models, and there's software out there commercially available where you can input this information and then do modeling on the particle size, how it will dissolve before you go out and start making batches of different particle sizes. So a very direct relationship between particle size and dissolution rate. This will be important when I start talking about the effect of not only dissolution rate but also bioavailability, and where we're dealing with poor solubility of certain drugs. A very small particle size down to the nanoscale becomes very advantageous. Another place where particle size is very important is content uniformity. If you imagine we have active ingredients here, if all of these particles are active ingredients, and if the smallest ones are the black ones, and then the green, the red, and the blue are as the particle sizes get larger, if this blue particle is too large, it has to end up in one of these doses, and then perhaps this dose has too high of a dose and it's not safe for the patient. So if you let particle size get out of control, then you start losing control of content uniformity. What you see down below is actually me showing this is not just a theoretical discussion. A few years ago, I actually just went onto the FDA website and searched for product recalls based on particle size, and I found these two products. And I'm a plate person. I don't show who the manufacturer was. But here's an oral suspension, and what you see the reason is due to large particle size. Some of the unit doses may not meet potency specifications. And here is actually a solid oral dosage form, and the same concern. The particle size range of the bulk active ingredient was outside the normal range, and we have trouble with content uniformity. So this is a very real concern, and one of the reasons why it's very important to track particle size when we're looking at specifically the active ingredient. But today I'm really going to be focused on smaller particle sizes. And what do you mean by smaller particle sizes? So I have a few slides to kind of show the scale we're going to be talking about. So if you just look at a water molecule, that's down on the scale of like 0.2 nanometers. If you look at a buckyball, that's just under a nanometer. But then when we start talking about proteins and some other biomolecules like an AT base pair, that's around the scale of 2 nanometers. Hemoglobin's on the scale of 6 nanometers. And what you see over here are silica nanoparticles, where if that scale is 50 nanometers, these are probably 20 or 30 nanometer silica nanoparticles. And we'll be talking about silica nanoparticles as a vehicle in a few of the slides. So really I'm kind of talking from a nanometer up to 100 nanometers, although we'll talk about slightly larger as well today. So here's this scale again, and on the scale we have a micron. 0.1 microns is 100 nanometers, so this is 10 nanometers, and this is 1 nanometer. So what you see down below is kind of some of these drug delivery vehicles, what size range are they in, and proteins might be on the scale of a couple of nanometers, maybe up to 10 or 20 or so. We'll be talking about dendrimers, which can be both smaller and larger. I'll be showing data of some polymeric nanoparticles, and those usually try to be down in the scale of hundreds of nanometers, but you'll actually see when I show some batches that went bad, that's scooting all the way up to 10 microns, which is a problem particle. Then liposomes really depends on how they're manufactured. They can be anywhere down from the scale of tens of nanometers up to, I've seen liposomes at 25, 30 microns. So that's the size range of some of the particles we'll be talking about today. And what you see up above is the technique we might be using. So dynamic light scattering can usually work from just under a nanometer to just over a micron. And we have the SZ100 DLS system. You'll see data today coming from this instrument. And our laser diffraction analyzer, the LA 950, can very easily work down to around, let's say, 30 nanometers up to 3,000 microns or 3 millimeters. And you'll see there's a crossover area in here where you could use either technique. And part of what I'll be discussing and showing data for is there certainly are nanoparticles where you could choose either or technique, and I'll be talking about perhaps how you might decide which one should you be using. Or in the best of all worlds, you probably need both, right? But for dynamic light scattering, that's the SC100. I just have one slide to talk about that. Right, so dynamic light scattering, this isn't a talk about dynamic light scattering. All I'm going to show is the one slide. The particles are here in a cell. We bring in a light source and we look at the Brownian motion. We use one of two detectors to track the Brownian motion, either at 90 degrees or in backscatter. And it's the only instrument that has that flexibility. It'll choose this automatically for you, or you can choose. And then we have this detector to measure the Zeta potential. We have the two positions. The cell can be right in the center there for low concentration, or here near the cell wall, higher concentration. So it's a very flexible tool for measuring particle size and for Zeta potential. The LA950 is the other instrument I'm going to be talking about, based on laser diffraction, which works, I usually say, from about 30 nanometers to 3,000 microns. Here the sample is going to pass through a cell. It could be wet or dry, but when we're talking about nanoparticles, they're wet. So what you see here is the cell where we have a reservoir and an ultrasonic probe. If we need to disperse the sample, it's recirculated through the cell. In the cell we have two light sources, and this is a diffraction pattern in the forward direction, but the technique does a lot more than just look at the forward direction. It looks at side scattering, it looks at back scattering, and takes all that information and calculates a particle size. So this is the other technique we'll be talking about, the LA950, which uses laser diffraction. I think that's all I say about instruments in today's talk. But we're talking about nanoparticles for drug delivery, and why nanoparticles? Why do we want them to be small? Well, part of it is because as we make these particles smaller, we improve the surface area to volume ratio. So there's more exposed surface. Since there's more exposed surface, these particles are going to dissolve quicker, and also the solubility. Maybe it works a lot better when it's in water, since it's a very small particle size. Since it is small, we improve the bioavailability. Sometimes we can get away with smaller doses, and there's less toxicity. We need to gauge the size just about right. We need it to be small enough so that it isn't picked up by the mononuclear phagocyte system. So it usually has to be around under 500 nanometers to avoid the MPS. And it also has to be large enough to avoid rapid renal filtration, meaning up over 3 to 5 nanometers. These small particles can cross cell membranes. They can interact with cell surfaces, and the other really cool thing that can happen is we can do targeting since we're talking about particles this small. There's a few ways to look at making these nanoparticles. There's the top-down, where we take an active ingredient that's a certain size, we pass it through some process, I'll talk of several processes, and we go from a large particle size to a small particle size in order to achieve the desired size. The other approach is bottom-up. This is just kind of showing how surfactants self-assemble into my cells. We actually use more complex self-assembly mechanisms for a lot of the samples we'll talk about today. But we have top-down and we have bottom-up techniques for making nanoparticles. I'll talk first about a few of the techniques for doing top-down. So this is actually the Elan nanocrystal technology, where it's a colloidal mill. We have milling media in here. We're recirculating the sample, and it's milled until we reach the desired particle size. To put a human scale on this is the mill sitting right here with these operators. And in the other room here, there's an LA950 tracking the process. And what they'll do is they'll mill for a certain period of time until they achieve the desired particle size distribution. And here are some results from that nano mill as measured on the LA950. It starts out at time t0 at about 48 microns, and then after 26 minutes we're down here into the scale of nanomaterials. This is at 155 nanometers. So this is showing off one of the reasons you might choose to use laser diffraction is if you need to track this size reduction, you need to be able to measure these larger particles and still be able to measure down to the scale of 100 nanometers. And the LA950 is quite capable of measuring across that entire range. This is other data from this particular publication, which also came from Elan. And this is actually, Ian will actually pick out, this is actually one of our older LA 920s, I think. But even the LA 920 was very good at measuring down to the scale of, this is about 150 nanometers or so. So this is showing an insulin product where they start out at 16 microns. They mill it using a particular surfactant at a certain pH, and they're just showing how they actually can mill it down to this range of 150, 180 nanometers. Another process where we have lots of data for reducing particle size down to the nanoscale is a microfluidizer. This is a technique which comes from our friends at Microfluidics. And here what we do is we have an inlet reservoir with large particles. We bring it up to high pressure, and then it passes through a chamber where typically we take these particle streams, split them, and then they meet again here under very high pressure, and we reduce the particle size down to a very small size. The samples I'm going to show that went through the microfluidizer, the first one I'll show is a liposome. When I started talking about self-assembling, I'd shown a micelle. This is a picture of a micelle here, where it's just simply surfactants self-assemble into a micelle. But if we take lipids, phospholipids, they can actually build more complex structures, such as this liposome. And what we can do here is we actually have hydrophilic and hydrophobic areas within the micelle. And what we can do is, I keep hitting my button there. I have to get back here again and take my arrow. There we go. So what we can do is we could actually hide a water-soluble drug, perhaps inside a liposome, which might be happier in an organic environment, or the other way around. So liposomes have been used for drug delivery systems for a while. Actually, I think Doxil is the first drug that was approved by the FDA that used a liposome as the carrier method. I think the FDA approved that back in 1995. So these have been around for a while, but the particle size is very important when we're using liposomes as our delivery mechanism. This is showing data of liposomes as it goes through a microfluidizer. And what you see, if you're familiar with laser diffraction data, this is the D10, the D50, and the D90. So we start with the D50 at 170 nanometers. And after we pass it through the microfluidizer, we bring it down to where this is on the nanoparticle scale with a D50 of 93 nanometers. So this could be measured both with dynamic light scattering and with laser diffraction. This is LE950 data because sometimes the initial product will be up over the size range where dynamic light scattering is highly effective. But this is showing the ability of laser diffraction to measure down here on the size scale as well.
Hi, Mark.
The next, yeah, hi.
We have one question about that last slide. So the microfluidics data, was that one passage or a number of passages?
This was a single pass.
Single pass. Okay, thank you.
Yeah, and we have this data, and Microfluidics is always happy to help share this data with us, and we appreciate that from them. You know, it can take multiple passes depending on what kind of product you're passing through there. A harder particle might take several passes. Since liposomes are fairly soft, usually that process doesn't take so long. If you're processing like a metal oxide, it probably takes a few passes. Polylactic acid, PLA, is a common carrier for nanoparticles. This is really kind of, there's some, an artist's concept of what a PLA nanoparticle looks like. It's not that this is the active ingredient here. So if the scale of the polymer particle is around, let's say up to 200 nanometers, it's not that these are 1 nanometer active ingredients. What usually happens is the active ingredient is interwoven into the polymer matrix. The delivery company then creates this matrix, creates a nanoparticle on the scale of 100 nanometers or so with the active ingredient woven into the polymer matrix. And then if we want to target the nanoparticle, we'll probably do something to the surface, some kind of ligand, so that this will go out and seek the area where we want to deliver the drug to. Usually it's a tumor or something like that. So there's a lot of research for this for cancer therapy, where what we want to do is introduce the drug to the patient, and rather than have it be systemically in the system for a long time, focused on the tumor region and apply the therapeutic payload to the tumor region. And there's also a lot of work on nanoparticles to be able to actually track the motion of these particles as well. I've seen data for nanoparticles that both deliver it and also act as tracer particles. Here we're probably looking at a fluorescence. Maybe these are quantum dots or something like that. So that we can not only deliver the particles, but also find ways to trace, was it delivered to the specific site. This is LA 950 data for PLA nanoparticles. This is a good batch, and what you see is these are already down in the scale of nanoparticles. We have a D50 at 81 nanometers, and this is a bad batch, and we have a D50 of 81 nanometers. And so actually this is why I want to visit this particular customer. This customer is a spin-off from MIT, one of the Bob Langer companies. And if you look at the D10, D50, D90, which is what most people do with laser diffraction, you'll see that these numbers are essentially the same. But if you look over here graphically, you actually see these few large particles, and these few large particles cause trouble, perhaps a content uniformity concern. So I visited this customer to talk to them about validation, method development, data interpretation, all the things we're very happy to help customers with. And what I suggested was, rather than looking at the D10, D50, D90, if you look at the volume mean diameter, that goes from about 82 nanometers up to 730 nanometers, a 9-fold increase. So we just talked about if you really need to catch the presence of these large particles, you can do it visually, but it's hard to write that into specifications. So you have to write a specification, then focus on the volume mean, or the D43, and that's probably somewhere we could actually catch this. And you know, on a lot of these topics, we have a separate webinar you can go listen to on setting particle specifications. If this is an interesting topic, that is something that's been recorded, and we have a lot of other information on, a very important topic, especially in the pharmaceutical industry. Now, this same customer recently sent us some sample of PLA nanoparticles, and we ran them on both laser diffraction, on the LA 950, and on dynamic light scattering using the SC100. Now, if you look at the Z average using dynamic light scattering, that was 90 nanometers. And if you look at the median size using laser diffraction, it's 80 nanometers. And this is what we typically see. And usually the results from dynamic light scattering will be larger than laser diffraction. We always say if you use two techniques, you'll get two different answers. But this is the trend you pretty much always see, and there's two reasons for that. Number one is when we use dynamic light scattering, we're looking at the hydrodynamic diameter. So that means a particle of this size diffuses at this rate. Now, if you have ligands or if you have other polymers hanging off of the surface of the particle, that's not going to increase the size of the particle very much, but it will slow down its motion. It's like running through the wind with your umbrella open, right? So it slows down the motion, and as you slow down the motion, it appears to be a larger particle. So that's reason number one. Reason number two is this is an intensity-based calculation, as opposed to laser diffraction is a volume-based calculation. And between the two, intensity distributions tend to look larger. So this is actually very good data and agrees fairly well, but we saw that for both batches of the sample the customer sent us. So here we have a Z average of 90 nanometers using the SC100. And on the next slide, this was a somewhat larger batch where we had 107 nanometer. The median by laser diffraction was 82. So a couple of things we see here is the same trend of we catch the larger particles. You get a larger result using dynamic light scattering. The differentiation's a bit larger, and that's because of these issues of hydrodynamic diameter and intensity-based distribution. So for this particular customer, what should they use? Well, they already have an LA 950. We think they should have an SC100. And partly we say that because we love people to buy instruments, but also in all seriousness, if you're working with small particles, you really wish you had more than one technique to measure particle size. A well-characterized lab, a well-stocked lab, has several techniques so you can compare one versus the other and make sure you have a very good feel for what's going on with the size of particles, assuming this is very important for the product performance, which in this case it is actually quite critical.
So we have a question asking what does the FDA like? Does the FDA have any kind of preference or something they like to see?
The FDA likes paperwork. So what the FDA will come ask you about is when you chose your technique, how did you justify it? When you made these measurements, how did you do your method development? Do you have the data proving you're using a method that's appropriate for this sample? The FDA is not going to say we prefer one technique or another. They're just going to make sure you did your due diligence and chose a technique appropriate for the samples you're measuring. And I know this, I won't say for a fact, but if you go far enough back in history, since your presenter has been in this business a long time, I was the first person to do particle training for the FDA inspectors. I had in Philadelphia, I'll say 12 years ago, and I met the people in the lab who looked at the data and sometimes asked to get samples to measure them. And I had a heart-to-heart with them about what do they really look at when they review data. I was kind of fishing more with laser diffraction. You know, do you check how people choose refractive index and to use Fraunhofer or me theory? And they said, that's actually a little more in depth than we're told to go. We're mostly making sure intelligent decisions were made and that we can review the data supporting the decisions made. Hopefully that's a complete enough answer. We can review it more later if you want. This is just going back again to intensity versus volume results. So, you know, 97 versus 86, that's at least the kind of differences you're going to see. Give me my pen back. So I apologize. So I need to bring my, oops, there we go, my mouse when I give these presentations. So I go back to here. I should just be talking while I do this. So we skipped back to the start. I apologize. You know, the reason why I didn't bring my mouse is because I'm flying on a cheap airline. And when you fly on a cheap airline, you can only bring one bag on board. So all I have with me on this trip is a backpack. And with my backpack, I was really limited with how much stuff I could bring on the plane with me. So I come down here to where we were. All right, so we were talking about hydrodynamic diameters, laser diffraction versus DLS. And we're talking about some other studies that I've seen where they talk about the difference between laser diffraction and dynamic light scattering. So this is data I found published in Pharmaceutical Research, where people actually published this particular active ingredient, and you see it's on the scale of 180 nanometers, both static light scattering, that's dynamic light scattering. You know it's DLS because it says Z average, and this is static light scattering. That's another name for laser diffraction. And in the data that was published here, you'll see, for instance, with dynamic light scattering, if you get 215 nanometers, the D50 is 130. And this trend of DLS always reporting a larger size is across the board. So if you ever are challenged by the FDA, you can refer to this particular article and say, you know, in this range, it has been characterized that both dynamic light scattering and laser diffraction is appropriate. So which should we use? Well, it depends on, do you need to see those larger particles, but also the sample volume. Dynamic light scattering, you can just get away with using lower volumes. You can get away with using lower concentrations. So sometimes it's a question of the sample itself. Sometimes you look around and say, well, for this particular product, what is typically published? And people use a technique which is typically published. Just be aware that when you're looking at these results, someone can take a DLS result and convert it to a volume distribution, and you need to make sure that the data you're reviewing is actually in a format that you know. This is actually data from an application note written by Jeff Bodycomb, where he was comparing some emulsions. And this is in that same size range of 150, 170 nanometers. And he's looking at the Z average from the SC100. So if the Z average is 120, when we convert it to a volume basis, it's probably 140. And you compare that to the D50 from laser diffraction, and all of a sudden we have a number that's a little more comparable, but still we're getting a larger size. So you also just have to make sure when you're looking at data from DLS, is it the Z average or on a volume basis, because all instruments have software that can do that conversion. Another point that you need to consider when choosing dynamic light scattering versus laser diffraction is, do you also need zeta potential? And if you also need zeta potential, then probably you're going to be using dynamic light scattering, because that's a technique that includes that information. Here are some batches, again, of some other PLA nanoparticles. Down here in the range where the D50 is 92 nanometers. And a customer tried to trick us on this one, and he actually said, I have a single, there was a single mode and then a bimodal. And he wanted to see if our instruments could really pick up on the bimodal. And the point is, this was actually spiked with 1 micron polystyrene, and you can see the LA950 actually did a very nice job picking that up. But also dynamic lights, the laser diffraction analyzer, this is the LA950 data, picked up on these large aggregates out here at 30 microns. And dynamic light scattering would just never see this. So this is the data from dynamic light scattering down here. And yep, we can pick up the 1 micron particles, but it's going to miss those 30 micron particles. So do you need to see those? Well then, perhaps laser diffraction is appropriate. If they're never there and you just need sub-micron sizing, that's where dynamic light scattering shines. We could look at a lot of data from colloidal gold or nano gold that's used for drug delivery. I'm going to show data in a few slides that was published in Langmuir, where I helped them run the data. And this is for a prodrug, right? So prodrugs are where the drug initially is in an inactive form, and then once it is delivered to the tumor cell, there's usually some kind of enzymatic reaction that takes place, and then it becomes activated. And so what we want to do is we want to have a carrier particle, and then we're going to have this prodrug, and we might have some kind of targeting device to help it find that tumor. In this case, the carrier drug was 50 nanometer gold particles. And what they did is they took these additional biomolecules and they wanted to attach these to the surface at different concentrations. They wanted to see what happened as a function of size as they were adding different concentrations onto the carrier particle. And we measured both the size and the zeta potential using the SC100. So the base particle is 51 nanometers. That's what the provider said. What we measured was 51 nanometers. They called it 50 nanometer nominal colloidal gold. We measured the initial zeta potential. And as you look at what's coated onto the surface, they should be probably around 5 nanometers. So if you coat it, maybe it gets up to 60. And what you see, and this is what was published in the article referenced here, is as we start coating, it goes up to 53, 56, it gets up to 60. And what we see is a less ordered and more ordered coating onto the gold nanoparticles depending on what we're coating the surface with. We didn't see big changes in the zeta potential, but we did pick up the changes as expected using dynamic light scattering for picking up the coating surface. So this is proving that dynamic light scattering has that ability to pick up what happened to my surface. That was the goal of that particular publication. We did measure the zeta potential of those particles, and this is not a zeta potential presentation. Hopefully we all know zeta potential is this charge at this distance from the surface of the particle that we call the slipping plane. And what we're typically doing is trying to create these stable dispersions. We don't want particles sticking together, or if it's an emulsion, particles phase separating. And what zeta potential is often used for is in dispersion stability. We'll modify the surface somehow. In this particular graph, we're modifying the pH, is this scale, and this is zeta potential, positive and negative zeta potential. And what happens is this is actually an emulsion where I did an isoelectric point study where if pH is 7, we have this nice single peak at a couple hundred nanometers, but if the pH drops down to 4, we reach the isoelectric point. The point where the zeta potential is 0 is the isoelectric point, and at this point usually the system destabilizes, and here the emulsion starts phase separating and we start seeing these larger droplets. That's usually what we're trying to avoid. So that's one use of zeta potential, is what happens with the surface chemistry and how do we avoid either this aggregation or phase separation if we're dealing with emulsions. So as we look at using this tool to measure isoelectric point, here's data which is the isoelectric point of a protein. This is lysozyme, which is a small protein. It's down at 3.4 nanometers. And this is just showing data I did a while ago of what happens as you change the isoelectric point, as you change the pH of lysozyme. And what happens is it's positively charged until we reach in and start increasing the pH. Then somewhere near pH 11, we actually go from positively to negatively charged. And as I pass through the isoelectric point, I could actually see in the beaker the particles starting to aggregate, because as they're very small, they're not even visible. And as we pass through the isoelectric point, they became visible. One of the great unique things about the SC100 is when we first were designing this, we actually had gold electrodes. And when we were measuring biomolecules such as proteins, they started to discolor. And what's happening here is because of Joule heating, we're actually kind of baking the sample onto the surface here. And that's not good. Then you don't know if you should really trust that data. And the cells aren't free. You have to throw it away and get another cell. So we switched over to a patented porous carbon electrode. And I've run many samples on these cells now. This is a study I did a year ago where I ran 800 samples using this particular cell. Still is working just fine. I remember I did this study and then I traveled to Budapest, where I am tonight, and started doing protein work at a university next door here, and it was working just fine. So that's one of the real advantages of the SC100. Being able to measure biomolecules, the zeta potential biomolecules, with a very simple procedure and a very long lifetime of the cells, which saves a lot of money in terms of cost of ownership. Another interesting use of zeta potential is it's just a way to understand what's going on the surface. So I show here an article which I'd found, which was published recently, and this is a customer in Japan who owns an SC100. And they were taking iron platinum nanoparticles, and then they were also using some silica composite particles to create a highly engineered particle where there also was use of PDDA, polydiallyl dimethyl ammonium chloride. And what they were looking at here is just what happens to size as a function of the concentration of the coating. And what's interesting here, so if we start out with some silica particles, as we changed the coating, what they saw was it was originally negatively charged silica particles were the template. And as you see here, as they started coating them, it went from negatively charged to positively charged. And this is how they knew they were actually successful in the coating, was the change in zeta potential. So you're not talking here about finding what happens, how does the charge reverse as a function of pH. It's as a question of concentration of the coating. And you can really use zeta potential in a lot of interesting ways to understand the coating of the particle, because what's going on the surface is what dominates what happens with zeta potential. So I thought this was an interesting use of zeta potential to understand the coating of these nanoparticles, which they're designing for use in drug delivery. So we went through some examples here. I guess it was my usual 45 minutes, right like always, Ian. We looked at data both from dynamic light scattering and from laser diffraction to study nanoparticles in this range that's of interest, down here 100 nanometers or so. We talked about when it's more appropriate to use laser diffraction, when is it more appropriate to use dynamic light scattering. You know, if everything's under 100 nanometers, you're probably using dynamic light scattering. Or if you have very small sample volumes or low concentration, you're probably using dynamic light scattering. But also, if you need to understand the particle-particle interactions and you're interested in the zeta potential, that's when you switch to dynamic light scattering, the SC100. But when should we use laser diffraction? Well, we should consider laser diffraction when we also need to be aware of the presence of those large particles. If those large particles really cause trouble with issues such as content uniformity, then you're going to catch those much better using laser diffraction than you would with dynamic light scattering. So that's the data I brought along today. The last point I'm going to make is, you know, if you go to our website and if you register and get into the download center here, for instance, I had mentioned there's a webinar on setting particle size specifications. There's a webinar on how to do method development for dynamic light scattering or laser diffraction. So we're just going to recommend, if those are other topics you're interested in, visit the website, make sure you register for the download center, and the past webinars as well as many application and technical notes are there. And if you register for the newsletter, then you'll make sure you're informed of when our next interesting webinar is. Did we decide where our next webinar is today, Ian? I thought we had that discussion earlier.
We did. So next week,
February.
Next week on February 20th, I'm going to be talking about how you know you can trust the size results you're getting. So you touched on that a little bit in your presentation about how everyone would like, ideally, orthogonal techniques, multiple techniques. So that'll be, let's see, that should be what, next Wednesday? Yeah, next Wednesday the 20th.
So we've actually gotten a bit better organized, and pretty soon you'll probably see on the website, you know, the upcoming webinars, so that you can choose which ones will be most interesting to you. But I thank everybody for their time today. That's what I've prepared, and I can see if there's any other questions that came up along the way.
All right, thanks very much, Mark. There were a few questions, and now would be a good time for those of you who arrived a little bit later. If you have any questions, go ahead and find the chat box and send them in now. If you need to run to lunch or to a meeting, or you have something else you need to do, I would encourage you all to complete the survey. So when you leave the webinar, you'll be presented with an option to take just a short 4 or 5 minute survey. Please do that. If for no other reason, then put in the comments area what you liked about the webinar, what you might think we can present next. You know, just any kind of feedback.
Mark, you idiot, bring
feedback.
your mouse with you. Little comments like that.
Especially comments like that. All right, so first question comes from Susan. She asks, how can you measure particle-sized droplets of very viscous or very concentrated products by process analytical technology?
Yeah, so PAT usually implies this is data that's going to be used to track the process of the manufacture of these delivery vehicles or the end unit, the end product. At the beginning, I used to think PAT always meant inline measurements, and that really isn't true anymore. It's just which measurements can you use to track the process. And 98% of all particle size measurements are still done in the laboratory, not in process. So if I'm understanding the question, first I'll say is it doesn't imply it has to be in line. Laboratory techniques are still appropriate. But these are highly viscous products, huh? And
or very concentrated.
These techniques aren't all that happy with that, and you end up having to do dilutions. You know, we talk about the SC100 being able to measure up to 40 volume percent, but that doesn't mean every sample you get the best results at high concentration. So with dynamic light scattering, you can try to measure at those higher concentrations. You have to know the viscosity to get accurate results. But if you're working at higher concentrations and you're using dynamic light scattering, you'd better be doing a dilution study to make sure that the results you're getting at higher concentrations still make sense. If you bring a high concentration, highly viscous sample up to a laser diffraction analyzer, you're always going to dilute it. There's no way around it. And you're going to dilute it and then get results. And what we usually do is we say, since the measurement is so fast, we don't think there's major changes in the size distribution, but out of those two techniques, the only warning I'm going to give is if you're tempted to use dynamic light scattering at high concentrations, make sure you do a dilution study. And if you read the ISO standard on using dynamic light scattering, it's going to tell you the same thing. Make sure you do a dilution study and that you're not seeing dramatic changes with dilution. When you do these dilution studies, it'll actually give you information of what's going on with the particle-particle interactions. But make sure you do that dilution study if you're tempted to reach for DLS.
So in summary, make sure you do the dilution study.
I could have said that quicker. Is that what you're telling me?
No, I just want to make sure. All right, so Antonio asks, for dynamic light scattering, is intensity a better basis than volume?
In general, yes. The calculation that is specified in the ISO standard, and I kind of go back to ISO standards frequently because ISO standards tell us what we can all agree to. I could have a different opinion of whether I preferred a volume or an intensity distribution. But in terms of what has been agreed upon by the international community, that's what shows up in the ISO standards. And the ISO standard for dynamic light scattering, there's actually two of them, but the most recent one and the older one both say what we all agree on is kind of how to reach a basic cumulant Z average calculation. So that is the majority of what you see published, and so that is what a lot of people use. Now, if I install a dynamic light scattering instrument, which I do, all the systems we sell in Europe, I usually go install them, then I'm looking at customers and I say, well, you get the Z average, but then we actually use a separate algorithm to calculate the mean. If you have more than one peak, then I usually say you probably want to switch to using one of the mean values, because the Z average doesn't give you any information about bimodal or multiple modes in your distribution. So what I usually say when I install instruments is use that Z average if it's a single mode. If it's bimodal, you're almost forced to switch to one of the mean diameters because that picks up on the modes of different peaks.
Okay. The next question is asking if there is a particle size at which the zeta potential is not always correct regardless of the pH.
So zeta potential is generally interesting for small particles because small particles have that high surface area where we have particle-particle interactions. Theoretically, you could measure the zeta potential of 100 micron particles, and I did that once and put it in a tech note just to say we can do it just like the competitors, so people can't use it as a lockout spec against us. But personally, I don't see interesting zeta potential of particles over 30 microns. The vast majority is under 1 micron. The place where it's no longer valid is when the particles are settling out when in the applied field within the cell. So the algorithms we use to dig the zeta potential information out of the cell is the motion is only due to the applied electric field. If the particles are settling due to gravity, then that is not modeled in how we make the measurement, and the data is in error. At what size do particles start settling? That's a function of both size and density. If you're talking about a very low density particle like latex, you can probably go up to 20 microns, maybe more. But if you're talking about heavy particles that are like a metal oxide, you'd better be giving up at about 2 microns. So it's a function of size, function of density, and as soon as you have gravitational settling, that result is in error.
Yeah, there's also a component too about scattering, right? So zeta potential is going to run into the same problem that dynamic light scattering will run into, where eventually you start running up against the issue where the particles get so small that the scattering off of them, the scattering signal becomes so weak that it can't be reliably detected. So generally it's true that if the particle is too small for the size measurement, it's too small for a zeta measurement.
Yeah, the lysozyme at 3.4 nanometers, no problem at all. I haven't personally worked with particles under 3 nanometers for zeta potential yet. If I had like gold nanoparticles that scattered really well, I probably could. I just haven't generated that data myself yet.
Good question. So Susan asks also, in an emulsion in an emulsion system, how would you tell the zeta potential of the emulsion droplets in a formulation versus the micelles that are also present in the external aqueous phase? So how would you differentiate between the zeta potential coming from the droplets in a formulation versus any micelles that might also be present?
Well, part of it is that issue of size and scattering that we just talked about. If those micelles are down at 2 or 3 nanometers, it's not going to have a big influence on zeta potential measurements. The emulsion droplets are going to be so much larger that's really going to give me the scattering. So it's something I haven't physically tested, and I usually like to respond to questions on personal experience. So I don't have personal experience on this, but that's the first comment I think of is if there's a way to just get the micelles separated, just run a solution up over the critical micelle concentration and see if you get a Zeta potential reading out of it or not. And if you don't, stop worrying about it, and your measurement's just coming from the emulsion droplets.
All right. Next question. For some of those nanoparticles, how do you get the refractive index? How do you know the refractive index?
Well, for things like polylactic acid, you can find the refractive index of polymers, not too much of a problem. So in general, there are means to find the real component of the refractive index. In terms of the imaginary component, we have a few webinars recorded on the subject, and we have a very, very good tool in the PLA 950 software called Method Expert, where it will do all these calculations for you automatically. So if you can come up with a value for the real component, let's say that's fixed at 1.6, there's a software tool in the PLA 950 software which will show you the results using imaginary values of like 0, 0.01, 0.1, 1, and 10. And it will pick out that best refractive index value for you by minimizing an error calculation. So what we generally say is literature or Google searches will usually point you towards a good value for the real component of refractive index, and use a Method Expert wizard to come up with the best choice of the imaginary value. But for a very full, complete answer to this question, visit the website and check out the webinar on how to select refractive index.
Okay, well, thank you, Mark. That was the last question. For those of you who are still here, I'm going to type into the chat box a general purpose email account at HORIBA for particle size. So you'll have my email address because of reminders that have come to you for today's webinar, and also you'll be getting an email saying thank you for attending, and here's a link where you can download the slides. But my last name, Trevoranis, isn't so easy to spell, so instead we like to give out labinfo@horiba.com. If you ever have any questions about particle size, particle shape, zeta potential, or surface area, please do shoot us an email. Otherwise, as Mark said, we have so much content on horiba.com/particle, so many webinars that have been recorded. I'm sure you'll be able to find hours of entertainment and knowledge there. And then you'll want to take a break and go get something to eat, like I'm sure Mark does right now.
That's the plan, Stan.
That's the plan, okay. All right, well, thanks to everyone for attending. As I said, the webinar should be recorded and up on the website within roughly 2 or 3 business days. Hope you all have a good rest of the day.
Okay, goodbye, everybody.
Bye.